Well, let me just give you a snapshot where we understand saturated fat. We're all humans, and we make palmitic acid. Are you surprised? Is anybody surprised with that? It's really interesting that wherever you go, we make palmitic acid all here. And you see, look at the percentage here. And I've spent most of my career in pediatrics. And so breast milk, or human milk, contains a fair amount of palmitic acid. So we should tell women not to breastfeed? That'd be a huge mistake, don't you think? Should we tell the bacteria in our gut to stop making palmitic acid? Because that's a primary intermediary for a lot of metabolic processes. And also, you see it everywhere. And interestingly enough, if you look at palmitic acid, it's kind of a staple throughout foods, all plants, all animals, all insects. Everything that's living has palmitic acid. So we stop living? Probably not a good outcome. By the way, those of you who like to take slides, you can have the slides. We'll give them to you. So pay attention. But if you want to take pictures, that's okay. But we'll give all the slides to you. And also, we look at the dominant fatty acids in palm milk and human milk. And babies need fat. Babies need energy. They're going through very rapid growth spurts. So, and obviously palmitic acid represents a very important component of breast milk. And lastly, we gave you some ideas where C16 is involved, and you can read that better than I can. And so does palm oil contain palmitic acid? Yes. But other fatty acids, all the other fatty acids contain palm oil, contain C16? Yes. But not all, as indicated in the last session, that in fact, not all saturated fats belong in the same bucket. So we want to talk about the buckets as we come up here. Dr. Pressman, I want to talk about this, please.
I'd just like to, thank you, Dr. Clemens. I'd just like to underscore the quality and the accuracy of our first presentation today, which really sets the stage for us. We're talking about saturated fatty acids in the most general sense, and in particular about palmitic acid. And to remind you all, obviously palmitic acid, C16, has as its major source palm oil. The reason that we're talking about this today is because palm oil has been under attack, palmitic acid has been under attack medically, and this is where it's a pleasure for us to talk about the evidence. This is not what we think. This is about reporting to you and communicating to you the preponderance of good evidence. And before we get into that evidence that says that palm oil is, as we've heard, absolutely safe from a nutritional and an atherogenic point of view, the evidence says that palmitic acid overwhelmingly is anti-carcinogenic. It is not pro-carcinogenic. The evidence says that palmitic acid is sufficiently protective of immunosurveillance and immunoresponse in the most general sense that it has the impact of some drugs. Now, having said that, let me very quickly remind you of something, and I have to do this as a physician. First of all, anybody who claims that any nutrient or any single factor causes a disease process is incorrect. Disease is very complicated and very messy. I think my colleague will agree that cardiovascular disease is multi-causal. As his wonderful slide suggests, we should not demonize any single variable here. An entire constellation of causation causes pathology. This is especially true for cancer. This is not a unitary phenomenon. This is not a unitary disease. There are at least 200 different diseases that we call cancer. There are many, many mechanisms that lead to the one thing that causes cancer, the one the one thing that all cancer cell lines have in common, which is immortality. Differences in tumor types, differences in tissues, differences in what interrupts normal apoptosis, differences in what messes up genes whose products either suppress or promote malignancy and metastasis and invasion. These things are very, very complicated. They are not attributable to a single element. Now, having said that, I don't think we need to gild the lily about how important palmitic acid is. We might mention that the evidence is that the dietary palmitic acid, or that palmitic acid in our dietary patterns, doesn't significantly enhance serum free fatty acids. So what enhances serum free fatty acids, by the way, is stress. What enhances urinary catecholamines is stress. The things we do to the animal studies that suggest that palmitic acid is bad are not due to palmitic acid. It's due to the stress we put on animals where we force feed them palmitic acid in a technique that many of you investigators know as gavage. It is simplistic and dangerous, again, to declare that palmitic acid causes pathology. Let's fly through some of these slides. I think we've covered much of this, and we've got the references if you would like to check us out here. Let's keep going. Pathophysiology of cancer, we just reviewed. So if you'd like to go back, if you have any questions about this, please contact us and let us know. Next, please. I think we can skip this, we've covered it. So we've covered, again, immortal cell lines, invasive and metastatic cells. The reason we're keying on this is because the two principal papers which we've referenced and which received global attention in the popular press declared that not only carcinogenesis in general was promoted, but that dietary palmitic acid created an environment at the cellular level which was conducive to metastasis or distant spread, in addition to invasiveness. This work was done in vitro, in cell lines, in petri dishes in a sense, or in well plates. There was a small study reported also using a very small number of animals, and this raises all kinds of questions about the validity of translational medicine. Incredibly important to develop our hypotheses, to ask questions, and to investigate individual pathways, but utterly inadequate to make any conclusions about human disease, especially, as I've noted, those animal studies were conducted by definition under levels of extreme stress. And we know that chronic extreme stress can cause inflammatory changes in many tissues, can cause inhibition of various immune responses, even in humans. We don't have to even limit this discussion to animal models, to rodent models or murine models. It has been amply demonstrated, replicated over and over again for years by Kikolt Glaser and their group at Michigan that natural killer cell activity and other principal elements of the immune system are significantly suppressed, even with short runs of stress of various levels. And that was operationalized well and documented well. Next slide, please. So again, we are questioning the validity of the research that claims that palmitic acid is this incredibly dangerous element. Again, absolutely not the case if we look at the preponderance of data. And we can look at our tables in which we've extracted a systematic review from a paper that was recently done that cataloged a number of studies in which palmitic acid was used specifically to target the basis or the variables that drive carcinogenesis. And it was demonstrated, as you'll see in the table, that in each case, the offending process was either attenuated or inhibited. Next slide, please. I think we can pass on that. Knowledge gaps. Dr. Hayes, would you like to take over and talk about where we're going with this?
Well, I think the big issue that we see is understanding what cancer is, and Peter mentioned there are at least 200 different types of cancers. And from a toxicological standpoint, when you think about cancer, it occurs really either in one of two general pathways. You have a genotoxic agent that causes a mutation, and that mutation is then copied and continue to be copied, and you get a potential cancer response. If it's not a genotoxic agent, which we have here, it's not a genotoxic agent, so it has to induce cancer by interacting with the endocrine complex systems, causing immunosuppression or autoimmune types of changes, or thirdly, to affect the immune system in some way, or cause hypertrophy, where you get cell changeover and you can now have additional cancer potential because of the mutations in the cell. So we don't see any of that in these studies that we've looked at. And so when we see in the context of all of this, we don't have any really basis for saying that there is a potential for this particular fatty acid to in any way cause cancer. Can we have the next slide? And here are some of the issues that one has to think about. As Peter mentioned, one of the studies was an in vitro study at extremely high levels, higher than you would suspect. Now, go back to that first slide that we showed you. The amount of palmitic acid that we actually have in the system, and particularly in newborns and mother's milk. So the dose is extremely important, and I just have to stop here. Our good friend put up a picture of a Hungarian, and he didn't give his name, he just showed us a picture of him. And his name is Paracelsus, or at least he renamed himself Paracelsus. And I don't know how many of you are well-versed in Greek history, but Celsius was a very, very famous Greek orator and writer. And so our friend that came up with that the dose matters and the dose makes the poison decided he would rename himself above Celsius, greater than Celsius, and that's where the name Paracelsus comes from. And so we have to be very, very concerned about the dose. These doses, even in the one animal study, were extremely high. Route, these were gavage studies where we're forcing large amounts of that material into the animal, taking a syringe and a particular type of needle, putting it down the rat's throat, hoping that we get into the GI tract and not into the lungs, and then sending a bolus shot. So we get inflammation just from that alone. The animal models we've already talked about. Probably the most important thing is we've not been able to show that there's any relevant or clinical significance to any of these studies. They don't translate to say this same type of reaction could occur in humans. And then the next slide, Do I have an Italian colleague that can read this for me? Are you Italian? Would you read that for me? Yes, and we cleverly took time to Google and how to translate it into your language. And do we have the next slide? I think, Peter, this comes back to you.
You know, it's interesting. If you look at how highly conserved palmitic acid is across both plant and animal species, it's almost as if we're talking about oxygen or water or carbon or any of these other things that we see as forever chemicals that are dangerous. These, I would argue, are forever chemicals, and palmitic acid is a forever nutrient that is actually the antithesis of poison. The other piece of this is, and the context is, that the dose piece of the weaknesses is very important here. If you exceed anything, it's likely to be bad. It's likely to be inhibitory. We're looking at a clear biphasic dose response, right? So at reasonable low doses, we get physiological levels and physiological effects, and we promote normal equilibrium and normal physiology. As you increase the dose, you end up sometimes with an opposite and even a direct inhibitory effect on multiple pathways. So the old common sense line here, the old saw about everything in moderation, is not entirely crazy. And we have to keep that in mind as well. The findings of the two papers that were written by these research groups that suggested bad things are interesting. It's interesting. It's very interesting bench science. And they focused on one or two or three hypotheses based on very discrete cellular mechanisms. That's fine. That's great. But again, understand not only the gross complexity of the development of disease and our responses to that insult, but you have to just, you must keep a very critical eye on simplistic, albeit satisfying, cause and effect relationships, because again, that's really not reality. Could we have the next slide? Oh, here we have some numbers which actually sets up a more detailed and specific discussion of dose here and the conclusions that were drawn on the basis of dose. I think Dr. Hayes had a very clear summary of that when we talked about this previously.
Yeah, I think that the real issue here is when you think about animal studies, you need to think about the dose. And this was a single dose gavage study. So we don't have a dose response. All we have is an overloaded system where we're artificially inducing something. And what was induced was a term that, can you go back one slide? One slide where nope, that's not it. They talked about what was the terminology that they used? No. Anyway, they coined a term in their paper, but didn't define what the term was. And it was some kind of pro-metastatic memory. And this was the basis for their conclusion, but they didn't define what they meant by this pro-metastatic memory. And the only thing we could figure out is that they're trying to say that somehow it kicks a response that will be remembered by the cells. But that was not clear in what they put out. And we point that out. What is the fact, what does pro-metastatic memory mean? And then they also had another term, anti-metastatic diet. And I think they were trying to say that the particular type of diet that they were feeding to these animals was an anti-metastatic diet. But again, remember, they were using very large doses, a limited number of animals, and they were giving it by the wrong route of exposure. And there was no dose response. There was just a single gavage dose at a high dose level. So if they'd done multiple doses, we could have seen whether or not there was actually a no observed adverse effect level in the study. So the study was poorly designed and poorly carried out. And we've talked about this, and our friend read it for us, and we've talked about this. The next slide. And I think that the real caution is the last, the level of the palmitic acid administered was by a stressful route, and it compromised normal effects of oral consumption, and we didn't really see any elevation in the plasma levels, and Peter already talked about that. And the next one, the next slide.
This is the table I was talking about, which I think is very important, but let me just mention one other general threat here. Anybody who's had experience at the bench understands that you can essentially take an extract of any compound, pick any compound, any organic compound, any extraction from a complex herbal or botanical, and make serial dilutions of that material and expose it to a cell line. Even expose it in various routes of administration and doses to an animal. By altering the dose, especially by enhancing the dose, as we've talked about, you can elicit a significant reaction in any experimental system that you want to completely unbalance. So you can produce, you can actually count on some significant measurable change in some sort of a metabolic product or any biomarker that you're measuring by applying anything to a simplistic model.
Let me just
Yeah.
take a minute. Most of you guys are nutritionalists. There's a word up here, apoptosis. Do you all know that word? Is that a word that's familiar to you? In the next slide, there's going to be a word, autophagy. Are those words that are familiar to you, or do we need them defined? You're good with them. Or you're not good with them. Okay, apoptosis comes out of embryology. If you look at your fingers, when you're in your mama's womb, you're webbed. And slowly a process of programmed cell death removes those. And so we don't have webbed fingers, say like a duck or something. Now, some of us, I have 2 webbed toes where it didn't go, you have 2 webbed toes too? Where the apoptosis didn't do its job, and so I have 2 toes that are basically webbed together. Now, autoplasia is a complex using the lysozymes. Programmed cell death is, this apoptosis is when the cell reaches basically the end point. The process says it's time to kill this cell. It's not necrosis, but it's apoptosis, planned cell death. Now, autoplasia is a little bit different. It's a process that our cells have to clean up junk that's in our cells. And there are organelles called lysosomes that have enzymes, and they combine, and we get this autoplasia, and the junk that's in our cell that we don't need is broken down, and we can clean up the cell. So I just want to be sure everybody understood those two terms that Peter is going to be talking about.
Yeah, so just to quickly come back to the research that comes down on the side of, at the very least, health promotion. We can look at the next three slides, in which we've simplified a table that, as I said, was derived from a huge systematic review that was recently published. So here you look at various mechanisms of abnormal cellular metabolism. So you have apoptosis here. So the problem with cancer cells is not that apoptosis occurs, it's that apoptosis does not occur. So these cells simply, as I said, don't die, they're immortal. So we have cells from Henrietta Lacks, who died at Johns Hopkins when? How many years ago? Is this 40 or 50 decades? 40, 50 years ago, 4 or 5 decades. Those cells are every bit as malignant as they were decades ago. These cells don't generally change. Proliferation is different, obviously, from apoptosis. This is not only that you don't die, but that you multiply, that you spread. So the data was cataloged to document the particular tissue type, the doses of palmitic acid that were tested, and the presumptive targets for palmitic acid action here. In every single case, and we could look at the next slide, metastasis, obviously distant spread, metabolic reprogramming, which probably involves epigenetic changes that are fairly significant, and then autophagy, another mechanism, as Dr. Hayes says, that ensures appropriate cell turnover. I think we have one more, Raj? Okay. So we've gotten to the conclusion here, but the take-home message from the table is that certainly we have no evidence from this collection of studies that anything other than a general suppression or inhibition of distant spread and malignancy has been noted in their model systems. We take this with a grain of salt as much as we take any investigation with a grain of salt. These are all studies that were apparently done by dedicated people in good faith at using the greatest technological means available. However, these studies generally suffer from the same kinds of methodologic threats that the ones we're criticizing suffer from. So, yes, the science is unsettled. It's not definitive and may never will be, and much of science won't be, because obviously that's the heady part of it. As we learn more, we refine and become more and more accurate and more capable of predicting what's going to happen tomorrow, which then informs our decisions to make things better. So that's the good stuff of science, as you know, and the bad stuff is that you got to take everything with a grain of salt, especially in regard to the handful of studies that have been done that suggest that things are negative when we're talking about palmitic acid.
I'm not a nutritional scientist, I'm a toxicologist, but what I've kind of learned here this week is that if you take too much salt, it's bad for you. But if you don't take enough, it's bad for you. If you take too much sugar, it's not good for you. But if you don't have enough sugar, you're in trouble. I see where this oil falls into that same category. We got to have this oil. If we don't get enough of it, we're in trouble. But it looks to me like it's even going to be harder to override palmitic oil than it is, say, with sugar or salt. Now, I may be incorrect on that, but that's the bottom line that I leave with, is that sugar and salt can be good or bad, and palmitic acid certainly is good, because there's a huge amount in mother's breast milk, there's a huge amount in small children, and there's a fairly sizable amount in each of us.
And of course, we do not consume palmitic acid, we consume palm oil. And as we've also learned this week, palm oil comes with multiple components, various bioactives, including tocotrienols and retinoids that are extremely important and obviously health promoting. So we are not saying at all here that the evidence is that palmitic acid is a drug. It's not a drug at all. And food is not a drug at all. But there are some commonalities. There's a little blurry zone here. Things like dose-response relationships, things like not drawing conclusions on the basis of a single element, a single variable. Remember, all these questions are multivariate. And discussions like this, debate like this, is extremely important because we see the weaknesses all around, we also see the strengths, and they point us toward the future. The future here is going to be actually on leveraging palm oil for other indications and other uses. And with that, I will turn you back to Dr. Clemens, and thank you very much for your attention. Ask us a lot of questions.
Thank you, Peter, and thank you, Wally. Do we have any closing remarks? You see, if you want access to the slides, just email me. I'll give them to you. I want this to be a positive learning educational opportunity for everybody. We want to encourage dialogue, either by that email or whatever works best for you. We want to encourage a continued education process. Any closing remarks from anybody? Sir?
Thank you very much for this concerned and concerning discussion, because it puts some doubts, and doubts is the source of science, I think. The scientific method is based on that. Oh, I'm a little bit, let's say, disappointed. The statement about models are at least naive. And, but actually, it's the only way not to run only with epidemiologists. Because the alternative is epidemiology, is statistics. It's making relationships, it's correlation. We need causality. We need causality. We need causality to understand mechanisms and to be sure that we are talking about the right thing and not just taking correlations by chance. But probably the right aspect is that we need the right model. So we need models, but we need also agreed, shared models. And translational models, of course. And another thing is about doses. It's not only acute doses, it's also chronic doses. In principle, I was a pharmaceutical chemist. Now I'm sold to the food science. But the pharmaceutical chemist knows very well that accumulation, because we have not efficient systems for detoxification for everything. So we may have some accumulation and some chronic effects. So probably we should also find models for chronic effects, not only for acute. If you agree, of course. And thank you very much for all the speakers today.
perfect remark. Thank you so much. Though I studied gastroenterology. I was a GI guy. And I studied at a little school in Boston called Harvard. But what is the best model for understanding GI function? The model system is the pig. And how many people study pigs? Though rats don't give us all the results, but they give us ideas because of model systems, and I respect that, absolutely. Any other great remarks, please? Well, there being none, we're going to transition over. Where's David?